Imagine unlocking the secrets of human life, understanding the intricate code that shapes our existence. This is the revolutionary impact of the Human Genome Project. Let's step back into the late 20th century, when scientists embarked on an ambitious journey to map the entire human genome, a project that would forever alter our understanding of human biology and disease. This groundbreaking endeavor, known as the Human Genome Project, was akin to decoding the very blueprint of life. In essence, the Human Genome Project was like unearthing a vast treasure trove of genetic information, a comprehensive guide to the roughly 3 billion base pairs that make up our DNA. Its completion marked the culmination of a 15-year-long international effort, a testament to the power of collaboration in the scientific community. The ripple effects of this project have been far-reaching and transformative. It has revolutionized our approach to medicine, paving the way for the rise of personalized medicine. No longer does one size fits all. Instead, we're moving towards treatments tailored to an individual's unique genetic makeup, a shift that promises to enhance the efficacy of therapies and minimize side effects. Yet, the Human Genome Project has done more than just transform medicine. It has ignited a flurry of debates about the ethical, legal, and social implications of genomic research. Questions about privacy, consent, and genetic discrimination have come to the fore, challenging us to navigate this new genomic era with care and responsibility. The project has also sparked a sense of awe and wonder at the complexity and beauty of life. It serves as a stark reminder of our shared humanity, that we are, at our core, 99.9% identical in our genetic makeup. But with this knowledge comes power, and with power, responsibility. As we continue to unearth the secrets of our genome and harness its potential for good, we must also grapple with the profound ethical and societal implications. And so, the Human Genome Project sets the stage for a new era in medicine and society. The Human Genome Project was not just an ambitious scientific endeavor, but a quest to decode our very being. This monumental task was set forth with a set of clear objectives, each as critical as the other. The first and perhaps most significant objective was the mapping and sequencing of the entire human genome. Picture this as creating the ultimate blueprint of human DNA, the very essence of life as we know it. This was a task of Herculean proportions. Remember, the human genome is made up of roughly 3 billion base pairs of DNA, the building blocks of life. So imagine the remarkable feat of not only identifying each of these, but also understanding their precise order. The second objective was to identify all the genes within this blueprint. Now, genes are like the instruction manuals for our bodies. They tell our cells what to do and when to do it. Identifying all the genes meant understanding the instructions that make us who we are, from the color of our eyes to our susceptibility to certain diseases. Scientists estimated there to be around 20 to 25,000 of these instruction manuals, or genes, in the human genome. The third objective was to store this information in databases, to create a resource that scientists all around the world could access and utilize. This objective was crucial in ensuring that the knowledge gained from the project could be used to advance our understanding of human health and disease. Finally, the project aimed to address the ethical, legal, and social issues that could arise from such a groundbreaking endeavor. This was a recognition of the profound impact this project could have, not just on science, but on society as a whole. These objectives, once mere aspirations, are now realities that are redefining the landscape of medicine. The Human Genome Project was a journey into the unknown, a quest to unlock the secrets of life itself. And, in achieving these objectives, we have taken significant strides towards understanding ourselves better than ever before. The Human Genome Project was a feat of scientific ingenuity, employing cutting-edge technology and innovative methodologies. To understand the methodology of the Human Genome Project, it's essential to delve into two pivotal techniques: DNA sequencing and bioinformatics. DNA sequencing, the process of determining the precise order of nucleotides within a DNA molecule, was the heart of the project. This monumental task was made possible by the Sanger method, also known as chain termination sequencing, which allowed scientists to read the DNA sequences. But the human genome is vast, with over 3 billion base pairs. To efficiently sequence this enormous amount of data, the project adopted a divide-and-conquer strategy, known as the shotgun sequencing approach. In this method, the DNA was broken into smaller, manageable fragments, sequenced individually, and then, like a colossal jigsaw puzzle, painstakingly assembled to form the complete genome sequence. However, DNA sequencing was just half the battle. The sheer volume of genomic data generated was staggering. To manage, analyze, and interpret this data, the project turned to the field of bioinformatics. Bioinformatics is essentially the application of computer science and information technology to the field of molecular biology. It provided the tools and techniques necessary to store, retrieve, organize, and analyze the colossal biological datasets. Through bioinformatics, scientists were able to create genome databases, develop algorithms for DNA sequence alignment, and predict the structure and function of proteins. All these processes were crucial in making sense of the raw sequence data, transforming it from a string of letters into meaningful biological information. Another notable methodology was the collaborative and open nature of the project. The Human Genome Project was an international effort, with scientists from around the globe contributing to the research. Moreover, the project committed to releasing the sequence data to the public domain, ensuring that the information would be freely available to researchers everywhere, thus accelerating scientific discovery. Through these pioneering methodologies, the Human Genome Project has unveiled the blueprint of human life. The findings of the Human Genome Project have been nothing short of astounding, revolutionizing our understanding of human health and disease. This ambitious endeavor has led to the identification of about 25,000 genes in human DNA. This knowledge, like a vast encyclopedia of life, is transforming our approach to medicine and healthcare. One of the key breakthroughs has been the association of certain genes with various diseases. This allows us to understand better the genetic underpinnings of conditions ranging from cancer to Alzheimer's. It's like having a roadmap of potential health risks, providing a clearer path towards prevention and treatment. This mapping of our genetic material is paving the way for personalized medicine. Imagine a future where treatment plans are tailored to your unique genetic makeup, increasing efficacy and minimizing side effects. It's like having a bespoke suit, only it's for your health. Furthermore, the Human Genome Project has spurred advancements in genetic testing. These tests can reveal an individual's genetic susceptibility to certain diseases, enabling proactive measures. It's like having a crystal ball, giving us a glimpse into our possible health futures. Gene therapy, too, owes much to the Human Genome Project. By identifying faulty genes responsible for certain conditions, scientists are exploring ways to repair or replace them. It's like having a mechanic for our genes, fixing what's broken or malfunctioning. Yet, these remarkable advancements come with ethical, legal, and social implications. There are concerns about privacy and consent, as genetic information is deeply personal. There's also the risk of genetic discrimination, where individuals could be treated unfairly based on their genetic profiles. It's like walking a tightrope, balancing the potential benefits with the potential risks. The Human Genome Project has not only reshaped medicine, but also raised profound questions about our society and our future. As we stand on the brink of this new era, we must navigate these complex issues with care, ensuring that the benefits of genomic research are realized while safeguarding our fundamental rights and values.